Thank you so much for the invitation and for organizing this fascinating conference. You're going to hear a lot of echoes of Allison's talk and my talk here. I think we have a lot of parallel ideas. So the title of my talk is Teenagers Are Not Lacking Their Frontal Lobes. And I realize I missed an opportunity for a joke here. Teenagers are so totally not lacking their frontal lobes. The fact that we can laugh so easily about teenagers, I think is really illustrative of a concept that we have about teenagers' brains. You know, just I say the word teenage brain, and it's easily the butt of a joke. So here you see a young man using his teenage frontal lobes. And what also is probably coming to mind for you are these other examples of what we consider inappropriate teenage behavior. Here's somebody driving, doing donuts. Here's somebody texting while driving, experimenting with drugs. And so this is true, despite completely awesome health during the teenage years. Teenagers have this large increase in morbidity and mortality due to engaging in risky behavior. Right, so this is a problem that as a society we want to fix, but I think it also leads us away from thinking about the incredible flexibility and creativity that teenagers have. And this is here where I'm echoing, I think, a lot of the messages of Allison's talk. And so here you can see creativity and flexibility in sport. And this fellow using his frontal lobes actually, solved an incredible problem there by getting up onto this planking mode. And the brain explanation for these differences in teenage behavior, we often go and point to the functioning of the frontal lobe. And there are actual scientific papers that compare the function of the frontal lobe in teenagers to the function of the frontal lobe in lobotomy patients or people that have suffered frontal injury. And indeed, there is some reason to support this kind of view of the frontal lobe as being under construction. The frontal lobe is still maturing into the teenage years. We already saw some data showing spine pruning. Here's also gray matter volume is decreasing 17% during the teenage years in the prefrontal cortex. And this word pruning refers to the synapses. These are the connections. They look rather twig-like that come out of neurons. And here you can see this is plotted on a log scale. Between 10 and here in the 20s and 30s, there's this radical decline in synapses and this pruning in the frontal cortex that occurs. So this is part of the reason for this brain basis saying that the frontal lobes are under construction. But really, are they functioning? And how do the brain changes support changes in function through this time? Now, when I started my own lab, this is a question that I wanted to study. We don't have great resolution to study synapses in the human brain, so we can move to mouse models to really study the neurobiology. And we can also study behavior in mice and try to correlate changes in the brain's function with changes in behavior. And so what I needed to look at the function of the frontal cortex in mice was a task which mice could perform very quickly, which mice could learn very quickly, that was sensitive to the function of the frontal lobe. And you can see here mice grow up. They have a very short childhood in comparison to the human childhood. They're weaned from their mother at P21. They're going through puberty around P30, and so they're P60. After P60, they're adults. One marker of adulthood that we have for laboratory mice and mice in the wild is when they become parents themselves. So we needed something the animals could learn in this teenage period, which is really just really P20 to P40, so the course of a month, which is not very long. Many tasks take animals weeks and weeks to learn, and they would be grown up by the time we finish testing them. So my graduate students went scouring the literature looking for tasks that were sensitive to frontal lesions, but that could be tested in just a few days. And we found one such task where the animals are foraging for Cheerios in an environment. So here they're just getting used to eating Cheerios. They're used to getting used to digging a Cheerio up when it's hidden under a bunch of shavings. And this is really when the task becomes dependent upon frontal cortex and frontal lobes, is when the Cheerio is buried in 1 out of 4 pots. The animal has to discriminate not the smell of Cheerio, but the smell of these distinct odor cues represented by colors here. There's a little bit of Cheerio smell in all of the pots, so they have to use this secondary cue to guide their foraging. And mice are, of course, using odors to forage in the wild, so they can learn this very quickly. In the space of an hour, they can learn that the licorice scent is where they should dig to find a Cheerio. In the course of another hour or two, they can also then learn to reverse this role. We move the placement of the Cheerio to a second odor. And the mice eventually give up looking in the licorice smelling pot and start digging in this, what is it, the clove smelling pot. Now, this is not a location-based task. We move the pots each trial. And we say they've reached criteria when they get 8 out of 10 consecutive trials correct. All right, so we were so excited to have this task and to begin to start monitoring when the mice had adult-like frontal function using this task. Now, we asked how do juvenile mice differ. This is a juvenile mouse. This is just after weaning. This is an adult mouse. Now, frontal lesion patients often show perseverative behavior or difficulty switching between rules or tasks, or difficulty switching from licorice to clove here. So if frontal cortex function is impaired in the juvenile mice, we would predict the juveniles would be slower than adults to reverse the rule from odor 1 to odor 2. So this is exactly what we expected when we started this task. And as often happens in science, the graduate student goes away and comes back and says, you know, it didn't turn out that way. So interestingly, the juvenile mice, when we ran this task, actually reversed faster than the adults. Now, there had been previous literature suggesting that juvenile or adolescent rats could look more like these lesion patients, so this was really surprising to us. We thought maybe these little juvenile mice just aren't using their frontal lobes to solve this task. So we went and we actually lesioned the frontal lobes in these mice. These are these areas of damage here in the frontal cortex, and they actually were using their frontal lobes. When they had the lesion, they were slower at this task, just as adults were slower. So this was lining up. So we thought maybe this is just an oddity for mouse development. But published back to back with our paper, there was another paper showing a reversal task in humans. Here on the y-axis is proportion correct, testing people as young as 10 up to age 25. And here you see this U-shaped function where higher bars are better. You can actually see from 13 to 17. In this particular task, the teenagers are reversing faster than adults or younger children. Here's another paper from Evelyn Krohn's lab looking at creativity and divergent thinking. And here again, you see that the 15 to 16-year-olds are showing behavior that's at adult-like levels or even superior to adult-like levels, superior to children. So when we look at the literature, when you have special tasks, maybe for the mice, our task actually had 4 different odors, which enhanced uncertainty in the environment. This particular task had very salient frowny faces, which was particularly stimulating potentially to, or motivating, prompting the reversal in the teenagers. So when we design our tasks the right way, we can elicit superior behavior. By our interpretation is going to be superior, and it does not look like, they do not look like lesion patients. So I think we've underestimated the function of the frontal cortex during this period. Maybe this is due to our interest in explaining poor teenage behavior, leads us to deeply underestimate the function of the adolescent brain. And I think what we need to do is update this model to imagine the adolescent brain is in a special period where they have differential decision making that's tuned for this particular life period. And also emphasizing a point that was made in Allison's talk, this period might be very important for exploiting new information and exploring the world, coming up with new strategies to live in a possibly uncertain or changing environment when the animals are dispersing from the natal environment. Whereas later in adulthood, we are more focused on exploiting the information that we've learned when we're settling down into a more stable environment. Here we have a burrow system and a cubicle system, where things probably aren't changing very much. But no, of course, mice aren't just little people. We have, of course, grandmothers and others helping us to figure out how to navigate this new and uncertain and broad world in humans. But mice are still exciting because they allow us to look at the biology of this at a deeper level. And so before I finish up, I just want to take you into a quick view into what's going on in the adolescent brain, which I think is also very stimulating. When we can focus in using imaging tools that we have in mice, we can look at the individual dendrites. Here is a postsynaptic neuron. We can think of spines as the little earpieces all over a neuron that are listening to information coming in from other neurons, coming from these synaptic connections made by nearby axons and boutons. What I'm going to do is I'm going to fill the postsynaptic cell in these dendritic spines with fluorescence and use a microscope to go in and look at them. And so here's a microscope focused onto an individual dendritic segment. This is like a branch of a tree of a neuron. Here's the whole neuron. We're focusing on the very top, the apical tuft. And you can see that the density of these spines goes down from the juvenile animal to the adult animal. So we see this pruning in mice in this postmortem tissue. But the very value of using mice in the last decade comes from new techniques like two-photon laser scanning microscopy. And the great thing about this technique is that we can look with cellular level, spine level resolution at the living brain. So the mouse can go under the microscope, the neurons get their picture taken, and the mouse can go back to the cage to continue to grow up. And we can go back day after day and see what the neurons are doing. And the insight that comes from these studies is really just a little over a decade old, is that we can see a new level of dynamics that is going on above and beyond that pruning that we know is happening. We see this daily turnover. This is a movie, this is actually the adult brain, so it's relatively stable compared to a juvenile brain. But these are the daily gain and loss of new spines on a neuron over the course of a week. And so you can see these new spines coming out. The rest of the background there is inky black, but there are many, many cells nearby, and there's axons of many cells nearby. And so we see these gains and losses occurring. And what we think is happening, here red would represent that postsynaptic side, that cell that I was just showing you that was filled with fluorescence. And what was in the inky black were nearby wires or axons of other cells. And everywhere an axon travels nearby a dendrite, you have this potential synapse. You can see in all these little circles, you can zoom in. And one of those spines reaches out about 2 microns, 1,000th of a millimeter, or 2,000th of a millimeter. It's a micron. And so these cells are reaching out and sampling new potential partners. And so they're exploring potential new connectivity. And now if we look at the data for adolescent development, you can see here between this weaning age and this adult age that on a daily basis, 25% of the connections are turning over. 25% are being gained and lost. So this is above and beyond that pruning that's happening. This is losses are outpacing gains, so eventually you get pruning. But this turnover is much more dramatic with this 25% change per day. And so I like to put this in context. I think you can think about the brain exploring all of these different possible wiring diagrams. It's kind of like exploring all these different potential possibilities of who they can be as a neuron. You know, am I going to be a neuron that cares about shapes or odors, or am I going to be a neuron that cares about really negative events or really positive events? And so this neuron is going through these radical changes, and then it's much more stable, and it's when it's reached its adult connectivity pattern, it's not going to change that a lot. So it's like this young woman here going through all her different costume changes. And here Diane Keaton has established a very stable pattern. She just makes very small changes in her style. So that might help you remember that data a little bit better. All right, so this again, I think, underscores the point that I'm trying to make, that potentially the brain is in a very different state at this time. And it's got this differential tuning and retuning that's going on, potentially to support this exploration of the possible state space and the possible individuals and the possible wiring diagrams that it can have in order to establish the right ones to fit into the society and to exploit as an adult. All right, and with that, I think just going back in hindsight, we go look back at where we started when we started these experiments and think, hey, this little mouse just ran out of food, and it doesn't have a lot of parenting help. Of course, it has a frontal cortex that's working quite well. It's probably in overdrive to make sure it gets all those calories at this time. So it seems kind of silly to imagine that the frontal cortex wasn't online yet, or was compared to damage at that time. And I think any parent says, of course my little darling is absolutely brilliant and creative and doing all sorts of amazing things. this is something that we apply to those bad kids or other teenagers. But still, any parent too might be also worried about, what can I do to help my child become an adult? What can I do to help my child become safe? And here I think the science also, it's interesting the different directions the science can drive us in. If we have this model of the frontal lobes being under construction and the child not having any top-down control, you really think about protection and basically locking the child up in your house until they're older and the frontal cortex comes online. But when you start thinking about that neuron that's reaching out and turning, the connectivity that's turning over, and the flexibility and creativity that's present at that time, I think about feeding more experience into this child, maybe even letting them fall on their face a little bit in order to learn and explore the world and basically develop their own rules for how they're going to deal with this. So again, I think this is great to go right after Alison Gopnik's talk. I think we have a lot of the same message. I'd like to thank you for listening, the people that did this work and my collaborators.